Bismillahirrahmanirrahim. Welcome back. This is part 2 of iron deficiency anemia. As we have discussed that iron deficiency anemia is a type of anemia where body stored iron is exhausted. Iron is present in stored form in our body in two forms: ferritin and hemosiderin. If an individual need iron and if the individual do not intake proper iron in its diet, our stored iron is released into the circulation for hemoglobin synthesis. Therefore, iron deficiency anemia developed in stages. Iron deficiency anemia is not an acute form of anemia, but it is a chronic form of anemia. Therefore, it takes time to develop. So there are three stages of iron deficiency anemia: iron depletion stage, iron deficient erythropoiesis, and iron deficiency anemia. We will discuss these three one by one. Iron depletion. As the name indicates that in this stage iron storage will be exhausted. Therefore, serum ferritin level will be decreased in this stage. Serum ferritin is in equilibrium with storage ferritin. So if storage ferritin decreases, serum ferritin will be automatically decreases. Therefore, in this stage, serum ferritin level is decreased, while serum iron is still normal in this stage. As the serum iron is normal, therefore there will be no anemia, because still iron is available for the process of hemoglobin synthesis, and if hemoglobin synthesis is normal, then no anemia will be developed in this stage. Erythropoiesis is also normal, therefore erythroid morphology will be normal in this stage. The second stage is iron deficient erythropoiesis, and the name indicates that in this process no iron will be available for the process of erythropoiesis. Therefore, in this stage, serum iron is also depleted, but anemia and hypochromia are still not detectable, because those iron which is transported into the erythroblast, that iron is involved in the process of hemoglobin synthesis, and still hemoglobin synthesis is a continuous process. Therefore, no anemia will be detectable in this stage. But as further iron is not available for the process of erythropoiesis, therefore only slight morphological changes will occur in this stage, and the erythroid cells, that is erythroblasts, will be slightly microcytic. The third stage is iron deficiency anemia, which is the advanced stage of iron deficiency. So in this stage, storage iron, serum iron is exhausted. No iron is available for hemoglobin synthesis, and hemoglobin synthesis is also affected. Therefore, hemoglobin synthesis decreases and anemia develops, which is known as iron deficiency anemia. This stage may be shorter if there is some additional abnormality like blood loss, like pregnancy. So these abnormality will shorten this stage, and iron deficiency anemia will be developed in increased concentration. Now, what are the causes of iron deficiency anemia? All those causes which affect iron absorption or which affect increased utilization of iron will cause iron deficiency anemia. For example, blood loss. Blood loss from GIT tract or menstruation blood loss or urinary blood loss and blood in sputum will cause iron deficiency anemia. These two causes, that is urinary blood loss and blood in sputum, are very rare, but still research have reported that these may also cause iron deficiency anemia, although it is very rare causes. Increased utilization of iron. Increased utilization occur during pregnancy, during infancy, during adolescence, and polycythemia rubra vera. In these conditions, iron utilization is increased. For example, in pregnancy, the fetus also required iron for hemoglobin synthesis. Therefore, more iron is transferred to fetus for development of hemoglobin synthesis. Therefore, the female developed iron deficiency anemia. In infancy and adolescence, more iron is required for growth. Therefore, iron deficiency anemia developed in these individuals. While in polycythemia rubra vera, polycythemia rubra vera is a myeloproliferative disorder where red blood cells production is increased. So if the red blood cells production is increased, so more iron is utilized in this process. Therefore, iron deficiency developed in these individuals. The other causes is malabsorption. Malabsorption occur in tropical stool, gastrectomy, and chronic atrophic gastritis. We will take one example like gastrectomy. In gastrectomy, stomach is removed. In the parietal cell of the stomach release HCl. HCl is responsible to convert ferric form of iron into ferrous, which is then absorbed to a specific receptor, DMT1 receptor. So if stomach is removed, HCl will not be secreted, and if HCl will not secreted, Fe3 form will not be converted into Fe2. Therefore, iron deficiency anemia develop in those individuals. Dietary inadequacy is another cause of iron deficiency anemia. In dietary inadequacy, those individuals who are strictly vegetarians, those individuals develop iron deficiency anemia. As plant source is a poor source of iron because plants contain phosphates and phytates which inhibit the iron absorption. Therefore, those individuals who are strictly vegetarians, in those individuals iron deficiency anemia develop. The other cause of iron deficiency anemia is parasitic infection like hookworm. Hookworm also causes iron deficiency anemia because hookworm sucks blood from the individual chronically. Now these are the signs and symptoms of iron deficiency anemia like fatigue, dizziness, headache, irritability, dry and pale skin, spoony shaped nails, which is known as colonakia, pica, and splenomegaly. Splenomegaly is very rare in iron deficiency anemia. Only 10% of the individual develop splenomegaly. The most important is that how we will diagnose iron deficiency anemia. So the first parameter is complete blood count. Complete blood count have these parameters like RBC count, hemoglobin, WBC, PCV, MCV, MCH, MCHC, and platelet count, and RDW. RBC count is decreased in iron deficiency anemia. hemoglobin is decreased, WBC count may be normal or increased. If there is parasitic infection, so WBC count may be increased. PCV, MCV, MCH and MCHC are decreased in iron deficiency anemia while platelet count is increased. Peripheral blood smear. If we make a slide and stain it with Romanowski stains like Leishman stain or Wright stain or Gemza stain or field stain and observe under microscope, so we will observe anisocytosis. Anisocytosis refers to variation in size of the RBC. If there are RBCs that are, some RBCs will be small, some RBC will be normal, some RBC will be decreased. So that condition is known as anisocytosis. In iron deficiency anemia, although the cells are microcyte, but in microcytes there will also be variation present. Poikilocytosis refers to variation in shape of RBC. If there are different shapes of RBC present on a peripheral smear, so we call it as poikilocytosis, like teardrop cells, like elliptocytes, like microcytes. So these cells are known as poikilocytes. Now this is a slide of iron deficiency anemia. We can see that this cell is microcyte, this cell is also 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 microcyte. These cells are known as microcytes. But there is variations among these cells, like this cell is smaller than this cell, this cell is smaller than this cell, similarly this cell is also smaller than this cell. So this is known as basically variations, which we refer to as anisocytosis. Now we can see that there are different shape of red blood cells, like this one. This cell is elliptical cell, so we call this cell as elliptocyte. This is also elliptocyte and this one is also elliptocyte because the shape of the cell is elliptical. So we call this elliptocytes, and if many cells are present, we call it elliptocytosis. If we look at these cells, the shape of this cell is look like tear, so we call this cell as teardrop cell. If you look at another cell like this one, we can see that this cell have a dot at the center just like a target. So this cell is known as target cells. Target cells are rare in iron deficiency anemia. So if different shape of red blood cells are present, we call it as poikilocytosis. Another thing which is most important is hypochromia. Normal red cells is 1/3 saturated with iron, but if we look at this cell is almost empty, so we call it hypochromia. Like this one cell is also hypochromic cell. So iron deficiency anemia is a microcytic hypochromic anemia. Reticulocyte count. Reticulocyte count is reduced in iron deficiency anemia. This is a reticulocyte. In order to see this slide, a special type of stain is recommended, which is known as retic stain or supravital stain. If we stain a red blood cell with supravital stain and observe under microscope, supravital stain basically stain RNA within a red blood cell. So this is basically RNA which is stained by the retic stain. And retic is immature RBC, which is not fully mature. Iron profile in iron deficiency anemia is also recommended, and on the basis of iron profile we diagnose iron deficiency anemia. So in iron profile the following parameters are usually included: serum iron, serum ferritin, TIBC, and transferrin saturation. So serum iron in iron deficiency anemia will be low, serum ferritin is also low, TIBC is increased, while transferrin saturation is low in iron deficiency anemia. TIBC, total iron binding capacity. So TIBC is known as that the capacity of transferrin to bind iron with itself is known as TIBC. As in iron deficiency anemia, transferrin have the capacity and the capability to bind iron, and there is no iron available. Therefore transferrin have the capability to bind more and more iron with itself. Therefore TIBC is increased in iron deficiency anemia. Bone marrow in iron deficiency anemia. This is the slide of bone marrow with a patient of iron deficiency anemia. If we look, these cells, these are erythroid series like this one cell, this cell, this cell, these are all erythroblast. If we observe a bone marrow slide under microscope, a normal bone marrow slide, so these erythroblasts will be, the size of these erythroblasts will be increased. But in iron deficiency anemia, the size of these erythroblasts is reduced to micro. So these cells are known as microblast. Another important thing is that in iron deficiency anemia, the cytoplasm of these microblasts is raked like this one. You can easily observe that there are small tiny projections present on the cytoplasm of these erythroblasts. Iron stain. Iron stain is recommended in iron deficiency anemia. If we stain a bone marrow slide with iron stain, we will see that iron stain and it will look like blue. So that's why iron stain is also known as Persian blue. So this is a normal slide of bone marrow which is stained through iron stain. But in iron deficiency anemia, as iron stored is exhausted, therefore this iron will not be stained and it will look like this. So there is a much difference, that there is no bluish stain, while this one there are a lot of iron. So in iron deficiency anemia, iron stain will be negative. Investigation that are occasionally required is stool examination for parasite. If we perform complete blood count and in complete blood count there is eosinophilic leukocytosis, we recommend stool examination for parasitic infections. Similarly, LFT, liver function test, is also recommended if there is additional abnormality present, like there is liver abnormalities present, so LFT is recommended, but these are occasional tests which are required. This is a slide of, this is a stool side, and this is basically the egg of an ova of hookworm. So in stool examinations, a hookworm ova will be seen that may cause iron deficiency anemia. Now differential diagnosis of iron deficiency anemia, that how we will differentiate iron deficiency anemia from the rest of microcytic hypochromic anemias. So iron deficiency anemia, chronic inflammatory RM malignancy, thalassemia trait, and sideroblastic anemia, all these are microcytic hypochromic anemias. So we can differentiate these three conditions from iron deficiency anemia on the basis of certain tests. So MCV and MCH. MCV and MCH are reduced in iron deficiency anemia, are reduced in anemia of chronic disorder, are decreased in thalassemia minor, and are decreased in sideroblastic anemia. Therefore, we call this condition as microcytic hypochromic anemia. Serum iron in iron deficiency anemia is reduced in anemia of chronic disorder. It is also reduced, but in thalassemia minor, serum iron is normal, and in sideroblastic anemia, serum iron is increased. So this is the one parameter on the basis of which we can differentiate iron deficiency anemia from thalassemia, where serum iron is normal, while in sideroblastic anemia, serum iron is raised. Total iron binding capacity, that is raised in iron deficiency anemia, while it is reduced in chronic inflammation or malignancy, or we can say anemia of chronic disorder, while in thalassemia minor, TIBC is normal, and in sideroblastic anemia, TIBC is also normal. So if a person have reduced level of serum iron and raised TIBC, so we can say that it is iron deficiency anemia, but it is also reduced in anemia of chronic disorder. Serum ferritin level is reduced in iron deficiency anemia, while in chronic inflammation it is raised. So we can differentiate anemia of chronic disorder from iron deficiency anemia on the basis of serum ferritin, where serum ferritin is raised in anemia of chronic disorder, while it is normal in thalassemia minor, while it is raised in sideroblastic anemia. Bone marrow iron store is absent in iron deficiency anemia, while it is present in anemia of chronic disorder, thalassemia trait, and sideroblastic anemia. Erythroblast iron absent, it is present, while in sideroblastic anemia it is present in ring form, which is known as ring erythroblast. So this is another differentiating point, that we can differentiate iron deficiency anemia from sideroblastic anemia, where in sideroblastic anemia ring sideroblast will be present. Hemoglobin electrophoresis is an important test which is recommended for the diagnosis of hemoglobinopathies. In iron deficiency anemia, hemoglobin electrophoresis is normal, while in anemia of chronic disorder it is also normal. But to differentiate thalassemia trait from iron deficiency anemia, hemoglobin electrophoresis is a main test on the basis of which we can differentiate thalassemia minor from iron deficiency anemia. So in thalassemia trait, raised level of hemoglobin A2 will indicate that this is thalassemia trait, because thalassemia is a hemoglobinopathy disorder. And similarly, in sideroblastic anemia, hemoglobin electrophoresis is normal. So these are various tests on the basis of which we can differentiate iron deficiency anemia from the rest microcytic hypochromic anemia. Thank you so much.